Hello everyone, this is Sukriti Joshi from Algerian Institute of Engineering and Technology. I'm going to take a video lecture series on chemistry. The subject code of chemistry is 311001. In our previous session of this particular subject, we have started our chapter number 7, which is chemical aspect of biotechnology. Chemical aspect of biotechnology involves various fields. That means we have to go through the knowledge of agriculture, food quality, as well as medicines. Along with it, we have also seen the manufacturing of ethanol also. Production of ethanol and production of acetic acid is a very important part of our chemical industry, because in general when we talk about its use, then we have to use it almost in everywhere. In medical field, propanol or ethanol are used as sanitizers. In chemical industries, they are used as reagents. In normal industry, or it will be used or it will be consumed as a liquor. Similar way, if we talk about acetic acid, then acetic acid is basically used or can be utilized in various types of food production units. In any kind of Chinese food, we use vinegar as a main ingredient. That vinegar is actually acetic acid. Apple cider vinegar forms or any other fruits vinegar gives rise to better gut bacteria. Even acetic acid is further utilized in various chemical industries for their own aspects. Like you all have seen that acetic acid acts as a reactant or reagent in that particular chemical reaction. Right? So this is why we have to go through this kind of things, and why fermentation plays a very important role. Right? Fermentation actually enhances the nutritional quality of any food. It has been observed that it can be seen that fermentation actually gives rise to better vitamin absorption, better nutrition absorption. So till now, we have seen that how we can use them into the production of particular ethanol from molasses. Now, in this session, we will see that how it can be produced from starch. Okay, so starch or starchy raw materials are generally used for the production of ethanol, like wheat, barley, rice, maize, and potatoes. Right? These are the materials which can give rise to the production of ethanol. Now, conversion of starch into maltose is possible. So, for conversion of starch into maltose and saccharification is carried out as follows. This particular process is known as saccharification because we need to produce saccharide from it. Okay, the first one involves malting. Okay, it is known as malt. So, moist barley is allowed to germinate in dark at 290 Kelvin. Now this germinated barley is called malt, and this is heated at 330 Kelvin to stop further germination, or otherwise it will get further germinated. Right? It is then crushed and extracted with water. This malt extract contains the enzyme diastase. Okay, diastase. Now, our next step is mashing. To break the cell walls, the starch is reacted with superheated steam. This exposes the starch. Inside that forms a paste-like mass called mash. And because of that, this particular step is known as mashing. Okay. Our next step will be hydrolysis. So mash and malt extract are treated together at 320 to 330 Kelvin. Now in about half an hour, hydrolysis is completed and maltose is formed. So these are the steps with which we can form ethanol from starch, right? Through fermentation. Let's observe it, that how it is possible chemically. So we will take starch, which is C6H10O5, present in N numbers. We have to take 2 molecules of it. We will react with N number of H2O in the presence of diastase, which is present in malt extract. Reaction will be done and we get maltose, which is NC12H22O11. Right? Here we get maltose. Fine? This is the chemical formula for maltose. And from that, as we have seen, the steps are there. From that maltose, we obtain our, again, ethanol. Now, let's talk about acetic acid. What is the meaning of it and how we can utilize it and how we can produce it. So, acetic acid, which is known as CH3COOH. Okay, this is the chemical formula, molecular formula of acetic acid, is also known as vinegar. Right, as I have already told you, this vinegar fermentation is one of the oldest fermentations that is known to man. That I should say that the fermentation process are the oldest ones, and from that the production of vinegar through fermentation is again the oldest one. Okay, man or human beings are started or have started producing vinegar in very early times, from very early times. Okay, it is formed naturally due to spoilage of wine. Therefore, literally, vinegar means sour wine because it is formed through souring of wine. So, it is known as sour wine. Generally, it is produced by reacting ethanol with oxygen in the presence of Acetobacter acetase enzyme. The enzyme which is responsible is Acetobacter acetase. Okay. Now, what is an enzyme actually? Right? Why we have to study about the enzyme? So, enzyme is a biological molecule which is used to increase the rate of biological reactions and even certain chemical reactions also. Here I can say that it is also used to increase the rate of certain biochemical reactions also. Right? It is used to produce biopharmaceuticals also. Let's take a look on characteristics of enzymes. That what are the main properties or characteristics of enzyme. First and foremost property is that the enzyme is a protein. The main material of enzyme is protein. It is made up of protein. It is made up of amino acids. Okay. Second characteristic is that it is easily influenced by environmental change. Simple environmental change and the characteristics of that enzyme will be changed. The role of that enzyme will be changed. So environmental factors include temperature, pH or inhibitor, any kind of inhibitor. If we change the temperature, if it's lower down, then the activity of the enzyme will be changed. If it is the temperature is raised, then the activity of the enzyme will be different. Similar way, if the pH changes, it gets deactivated, or at certain pH changes, it gets activated. Likewise, inhibitors, under the influence of inhibitors, the enzyme can be inhibited also. Okay. Then we have the third and most important one, which is it acts as a catalyst. Okay. The enzyme functions in accelerating chemical reactions, but the enzyme itself does not change after the reaction ends. Itself it will not change, or it will not create any kind of change in any kind of biological reaction or any kind of chemical reaction. But it will raise the rate of reaction in it. It will increase the rate of reaction. Okay. Now, next is the next characteristic, which is known as fourth one. It functions specifically. Okay, so the enzyme only catalyzes one kind of substrate and cannot function for many substrates. The term is called one enzyme, one substrate. For one substrate, only one enzyme will be worked and none other than that will work for it. Right? This is the thing that you need to understand, that for production of acetic acid, certain enzymes will be used. For production of ethanol, certain enzymes will be used. It is not like that you can use any enzyme for any kind of reaction. Okay? It is very specific. Now, let's move ahead to the next one, which is fifth. So it functions in reverse. It means the enzyme does not determine the direction of a reaction, but it only functions in accelerating reaction rate until it reaches equilibrium. Right? The enzyme also functions in substance synthesis and substance breaking down reactions. It can play a role in any kind of reaction. It is just like that. It works specifically. Okay? Now, next is, it is required in very small amount. That means a small amount of enzyme is able to catalyze a chemical reaction. Fine. Our next is biofertilizers. So, we will take a look on different applications of these things. From that, first is biofertilizers. So biofertilizers are the substances that contains microorganisms living or latent cells. Biofertilizers generally increases the nutrients of host plants when applied to their seeds, plant surface or soil by colonizing the rhizosphere of the plant. Biofertilizers are most cost effective as compared to any kind of chemical fertilizer. So here, let's understood what does it mean. Bio means life, and therefore by definition, biofertilizers are living organisms that enrich the nutrient quality of the soil. It refers to the use of microbes instead of chemicals to enhance the nutrition of the soil, as it is, or as a result, it is also less harmful and does not cause pollution, right? It reduces the soil pollution. This is what makes them so important to organic farming, because they are completely environment friendly, right? Types of biofertilizers involve bacteria, fungi, etc. Right? There are n number of things. Now, next application is biofuels. Biofuels are non-fossil fuels. They are energy carriers that store the energy derived from the organic materials, generally from biomass, including plant materials and animal waste. Right? They may be solids such as fuel wood, charcoal, and wood pellets, and liquids such as ethanol, biodiesel, and pyrolysis oils, or gaseous such as biogas also. Our next application is bioreactors. So bioreactor is defined as vessel that contain or that carries out a biological reaction and is used to culture aerobic cells for conducting cellular or enzymatic immobilization. Right? They are very important for us nowadays. Right? Next is biosurfactants. So biosurfactants are the surfactants of microbial origin. They offer so many advantages over their synthetic counterparts due to their biodegradable and environmental friendly nature. That's why gaining much more attention in creating the era of green technology. These are the applications, these are the chemical applications or chemical aspects of biotechnology. In different fields, nowadays it is possible that it is utilizing each and every field which surrounds us. Okay, this is all for this session. Next we will see in our next session. Thank you so much, everyone.